case study for one of the first, or actually the first FDA-approved drug for melanoma. So what we're going to focus on is first understanding the general mechanisms of cancer and how tumors develop and metastasize. And then we're going to identify the key metabolic pathways, and there's only a few, and how we determine from these pathways which proteins and enzymes involved are drug targets. And then we'll understand really the workflow of how cancer drugs are discovered, whether that be, there's a lot of structural involvement. There's a lot of, you have to look at it from every angle, right? Everything from the X-ray structure to the metabolism, as we know, the pharmacokinetics. And then understanding the difficulty of cancer treatment, of why it's very difficult to treat cancer. So we will get started. So cancer is driven, and like on these slides, I have the main bullet point is the title. Cancer is driven by the accumulation of oncogenes that lead to unregulated cell proliferation. That's cancer in a nutshell. So over time, right, over your lifespan, your natural cell cycle mitosis in every one of your tissues will develop mutations. Most of those times, those mutations are weeded out. Those mutated DNA strands are either fixed by different cell cycle checkpoints, like the CDK cyclin mechanism, right? By P53, which activates that mechanism. P53 is a very important protein that is, we call it, it's a tumor suppressor gene. And one of the codes for the P53 protein that allows these cyclin CDK checkpoints to work and to detect DNA damage and either cause apoptosis, meaning a programmed cell death, or to repair the DNA, which is also possible. So there's like a methylation, right? You can repair that by just taking out, doing an endonuclease, popping that nucleotide out and replacing it. That is an enzyme system to do that. So, but so the question is that if we look at these numbers, that if one mutation happens every 1 million cell divisions, by the time we reach adulthood, we have 10 to the 26 cell divisions. So the question is, why doesn't everybody have cancer, right? Because that seems like we're going to develop a lot of mutations. And the answer is apoptosis. We have cell cycle checkpoints and DNA damage repair mechanisms, which we're not going to cover in super detail, would be here all day or all night. But these mechanisms are in place to either repair the DNA damage or to just say, look, the damage is too far gone. We're going to cause the cell to go through apoptosis. Also, these mutations, when they relate to cancer, they have to be very specific mutations and what we call driver mutations. That's the last bullet point right here. Driver mutation accumulation with age. So as you get older, there are specific mutants that are really bad that can be a main driver in cancer development. So a lot of these are KRAS, right? KRAS was the first one discovered. There's BRAF, there's PI3K, there's P53, there's this protein called APC, there's WNT, that's in more colon cancer, and there's a bunch more that are key mutations that, when they become mutated with whatever amino acid, it can be really bad because they cause a change in function of the normal cell pathway. So it's not only one mutation. So let's say like there could be a chance one of us in this room has a BRAF mutation in our liver, right? But it's not just one, I hope not. But it's not just one of these mutations that causes the cancer. It's the accumulation of several of them. So if you have a KRAS and a BRAF wouldn't happen. They're in the same pathway. But if you have a KRAS and a P53 mutation, chances are you're going to get cancer in that tissue. And what happens when this cancer grows, it creates a tumor, it creates unregulated cell growth, meaning you don't only have this one pathway that's causing the cells to grow, but since it's the accumulation of the mutations, you have multiple pathways in the same tissue type that are causing this cell growth with no regulation, so no negative feedback, which is the dysregulation part. So this can happen and it can create tumors. The tumors then grow. The cells then break off and can go into different tissues. And then what happens when the cells get lysed, these proteins go into your bloodstream and they can enter different tissues. And then you can end up with metastasizing cancers, meaning those mutations can go elsewhere in your body. And they may mutate even further. So whereas, let's say, a Wnt mutation, that's a protein in the stomach, is in the colon lining, that mutation is not prevalent anywhere else. It's not prevalent in leukemias, for example, right? But there are, I'm sure there's KRAS mutations in leukemia. So it might cause the production of cancer cells in a different organ, but then those cancer cells might mutate into KRAS mutants. So this, over time, makes cancer extremely difficult to treat. You may be treating someone with colon cancer, where it's like, oh, okay, we have this regimen. We could use this inhibitor, and this inhibitor to cure your colon cancer. But then they go into remission and everything's okay. A year later, they have cancer in their liver now. Why is that? Well, because during the colon cancer time, there was some metastasis going on, meaning spreading of cancer to different tissues. And that was not really noticed by the doctors. Usually they catch it early, but it wasn't really noticed by the doctors until a year later, where they went in for another check and they see there's a tumor in the liver, right? So that's why it's extremely difficult. And also, that tumor in the liver, it's not going to be the same cells as the tumor in the stomach. So you can't just treat it with the same inhibitor. It might have a different genetic makeup. So that's what makes this extremely difficult to treat. Yes. Yeah, exactly. So skin cancer, yeah. Right. Well, the mutations would. So that's more so for those of you online, the question was about skin cancer and old age, because people who are older have more sun exposure, more UV exposure. So that's an epigenetic factor that causes cancer. So epigenetic factors, we talk about environmental factors, like UV radiation can directly damage DNA. And we know this from years of studying, that if you have a prolonged exposure to UV radiation for X amount of years, your chances of developing this kind of skin cancer is extremely increased, because over time, not only the cell mechanisms or the cell checkpoints break down, but you're also forcibly developing those mutations by a prolonged exposure by UV radiation. Yeah. Yes. Yeah. So telomere shortening, that's a really good point. So that's what happens after every round of replication. Your telomere, which is at the end of the DNA, gets a little bit shorter because of the 3' overhang, right? And this can shorten the DNA and then eventually mutate some proteins by leaving out certain sequences that would normally code for a protein or code for a normal protein, which now code for a mutated or no protein. So yeah, it's a very good point. So all of these put together increases the chance of mutation. Yes. Free radicals, yes. So so exactly, the question was about free radicals. Now, free radicals can cause the DNA methylation. They cause the DNA damage, which then leads to mutated or no protein. So, or usually mutated protein, which then leads to the cancer. So, like hypermethylation is one example that can be caused by like oxygen radicals, right? So that's why it's always good. Sorry, I'll get to your question in 1 second. But that's why it's always good to decrease your amount of free radicals in your body, right? And what do you take for that? Antioxidants. That's why it's really important, and it's been stressed over the last 10 years or so, to eat foods or take pills that have a lot of antioxidants that quench those oxygen radicals in order to preserve your DNA. Yes. Oh, I'm sorry, can you repeat the last? Proton therapy? Yes. Is it mainly for specific types of cancer? I'm not exactly sure. That's something we can look up. But I've heard it used for several cancers. I'm not exactly sure which one is it mainly. Yeah, yeah. Okay, yeah, I definitely heard that, but I'm not sure if it's just for those. But I'm assuming there's a wide range of variability for that treatment. So that's something we can look up. Yeah, Daniel. Malignant, because so good question, because they they stop growing. So what happens is after a certain time, there might be some regulation mechanisms that from, let's say, neighboring cells or neighboring organs that do stop a mutation in those new organs. So let's say if the benign tumor has just a KRAS mutation, right? But the organ that's surrounding that tumor, the cell growth of that organ does not really rely on KRAS pathway. It might rely on another pathway that doesn't really involve KRAS. So even if there was a KRAS mutant, it wouldn't really affect the growth of that cell. Therefore, it wouldn't be cancerous in that cell, in that tissue type. That's one example. But yeah, good question. Okay, so all good stuff. Let's move on. So here's just the evolution of the different genes that were identified that cause cancer, and that are these key driver mutations. And you can see there's a ton. But there's KRAS was the first one in the 80s. Then after 2003, so what happened after 2003? Anybody know? In a big deal in the science world? Yes, Human Genome Project. So after the Human Genome Project, a lot of these genes were identified that have a lot of implications in cancer. So 2004, we have EGFR. So EGFR is in the same pathway as KRAS and MAP kinase pathway. 2009, we have a bunch more. We have BRAF, we have HER2, we have PI3K, and the CA is just different types, a different isoform. ALK, that's a kinase. So yeah, but BRAF, EGFR, and KRAS are all in the MAP kinase pathway. That's why I focused my thesis on it, but also that's why it's the most prevalent target in cancer drug discovery for a pathway. And then 2014 is a bunch more. So yeah, basically, the more we know, the more cancer mutants we can determine. So here is what these pathways look like. And don't worry, I'm not gonna test you on rewrite this, right? It's crazy. So there's a lot going on, but generally speaking, there's two main pathways that contributed to 90% of cancers, or 90% of these mutations are in this picture right here, with the with MAP kinase and the PI3K AKT pathway. So on the left, in the like kind of neon green, we see the PI3K pathway, and we're not going to cover it in super detail. It's very similar to what we covered in the signal transduction and receptor lecture. So you have a ligand that binds to a receptor, this EGFR on the top, right? This type of receptor is, well, how about this? Can anybody answer the question of what type of receptor this is? You should know the top 5. Not G protein coupled.
Is that a tyrosine kinase, Professor?
Yes, it is. Good job, Ben. So yeah, it's RTK, right? Receptor tyrosine kinase, which means it has the dimer that the EGFR binds. They dimerize, and they autophosphorylate. So that means they create phosphates, which then could be linkers to other adapter proteins such as GREB2 and SHIC. Those can then lead to other recruitment of other proteins like GAB1, and then eventually PI3K, and then it can go down the kinase cascade of, you see all the P's, or that's all stands for phosphorylation, phosphate. So those phosphates are introduced that can either activate or inhibit the drug, or inhibit the protein, which then lead to cell cycle activation or just gene expression in the first place. So, and that's what leads to it. So you have PI3K, and then on the right side in the blue, you have the MAP kinase pathway, which you have SOS as the main activator and activates RAS, which has a lot of downstream effects, which not only limited to what's shown here, and then again, a lot of kinase cascade effect, which leads to transcription and translation. So yeah. So any questions on that? No? Okay. So I did mention before that these mutations that cause cancer they can change the function of the pathway, and they can change the function of the entire cell, right? There's two ways it can do it. It could either be a gain of function or a loss of function. And remember one of the, maybe it was 2 or 3 weeks ago, we were talking about just general drug discovery. Oh no, it was a receptor lecture again, where we mentioned that there are two things that we design a drug to do. The first is to stop a bad thing, and the second is to start a good thing. Those are the only two, in a nutshell, those are the only two things, right? This is the same thing, but with cancer, right? With cancer, you can only have two types of mutations. So think about the mutations as being bad, right? Mutations are bad. So what would the mutations that are bad want to do? They want to start a bad thing or stop a good thing. That's exactly what these types of mutations do. So a gain of function, right here, gain of function. That means we are starting a bad thing. So we are starting KRAS and BRAF, which means they are going to be consistently active, or that word is constitutively. And that's actually, I love that word. It's great. Hard to say, hard to spell, but it's a good word. Constitutive. So constitutive. No, it's constitutive. But anyway, good word. So they can be constitutively active, which means they're perpetually active. Why don't they just use perpetually? I don't know. Some papers use them interchangeably just to sound more fancy, but it means the receptor or the proteins were perpetually active, which means that going upstream and looking at EGFR, you don't even need this EGF, extracellular growth factor. You don't even need EGF to activate this pathway if you have something internal that's constitutively active, meaning whatever mutation it has, it's always in the on state and nothing can stop it. Even the negative feedback loop sometimes may not work. So this is an idea of a gain of function, adding or starting a bad thing. You can also have stopping a good thing, otherwise known as loss of function. If you stop a good thing, that's the opposite of what we're trying to do as drug discoverers. So that creates a problem. In this case, that problem is cancer. So we're trying to, or these cancer mutants are trying to lose the function. So lose the ability to stop growing. So in other words, start growing. Lose the ability to stop growing, start growing. You have the apoptotic mechanisms. It's like the caspases. You can have cyclin, CDK cyclin pathway, P53. All of those are subject to these loss of function mutations. And for P53, it's very interesting. Is that even? I see RBE10. Where's P53? May not be shown. Where? Oh no, but P53 is somewhere lurking around. Somewhere. But anyway, P53 doesn't directly have to be mutated for this to be a driver mutation. It could be this RB. What is it? 27. The regulation of P53. So it's not only the protein of interest that we're talking about, P53, that has to be directly mutated in the cells. It could be a protein that modulates that protein. That if that one's affected, it creates cancer. And that's another reason why it's difficult to treat. So imagine you have a patient that has two of these problems. Their P53 is all messed up, but they only have, or they have a mutation on P53 itself, and also on, what are you going to treat? You might treat both, maybe. You might, if you treat one, the other one, you really can't treat it at all. Or you might treat both, and it might be there might be a drug interaction with those two drugs, or they might have severe side effects, right? It might be nausea, vomiting, like a lot because of those drugs, but they might work and they might not work. Who knows? So there's a lot of these cases where, and we're still learning, because there's so many chemotherapeutics out there, that if we find individualized patient genome profiles that have certain mutations but not other ones, or multiple mutations, which most of them do, they are extremely difficult to treat because of this. You might have drug interactions or really, really bad side effects. So yeah, so I would say for you guys, for the exams and everything, just know that what are cancer cells going to do? Start a bad thing, stop a good thing. What is this called? Gain of function and loss of function, mutations. All right, so the case study that we're going to be talking about today is skin cancer with melanoma. So skin cancer, like we mentioned, can be caused by different factors, right? And one of these factors is epigenetic, which means UV ray exposure. And the melanoma is mostly in the epidermis. So it's really the top layer of the skin cells. And we have different genome profiles of expression, different expression profiles on different stages of the cancer. That's something I didn't mention yet. But in different stages of the cancer, because of the time that one of these mutated enzymes was upregulated, or when it was active, it upregulates or downregulates, basically throws out of whack the gene expression for that cell. And that is another driving force of metastasis. That over time, we have this changing of the cell function. So in different stages, so like if you have a senescent, let's say, so going from the beginning, so normally you have these little purple cells, right? Then you have this case where you have BRAF, the V600E. So you see the little V600. You know what that is? What is V600E? You don't have to tell me exactly what it is, but what does it represent? No. What is it? No, not the frequency of light. Good idea though. So it represents the amino acid. So what it represents is that the valine that was normally at 600, residue position 600, is now mutated out for a glutamic acid. That's what E is. So that's what that V600E stands for. Whenever you see those little numbers and letter next to a protein, that represents the mutation that it has. So, yeah. This P16 CDKN2, that is not a mutation. That is a isoform of it. Yeah, so it's tricky, you know. But if you see letter, that's a mutation. So anyway, these proteins become upregulated when cancer starts. Then the P16 becomes downregulated, but other ones also become downregulated, like CDK becomes downregulated. Over time, this is really bad. CDK is important for maintaining our cell cycle checkpoints. So over the progression of this cancer, that system becomes degraded. Then BRAF V600E is still increased. Then the amount of what this does also is it increases through the mechanisms. It increases the amount of PI3K. It might be normal PI3K, but the more PI3K you have, the more cell proliferation you have. It has its own pathway to increase cell proliferation. So basically, what this cancer is doing, and what a lot of cancers do, but what this melanoma is doing, is because you have one mutation over time, cell proliferation of BRAF has an interplay. Negative feedback of this one. So we'll try one example. And the negative feedback of this one. The monocytus pathway will be something in the beginning might be phosphorylate to turn it off. If that happens, it doesn't matter. With BRAF, it will always be constitutively active, right? So it doesn't matter if we go through these negative feedback loops. We'll still activate. It will still continue to be active. But the cell doesn't know that. So the cell will think of these negative feedbacks. We'll then think that because of these negative feedbacks, we need to transition to other pathways. for growth, because we're not getting MAP kinase growth anymore because we just did the negative feedback. But the car is still on, you know, it's still active because it's constitutively active. So that then has a crosstalk and activates the PI3K pathway, upregulating the amount of PI3K, because the cell thinks it's no longer growing, but it actually is. That's what creates even more cancer, and that's what starts the metastasis, which then can lead into different tissues, etc. So yeah, so that's basically what's happening. And then a lot of other stuff is upregulated too. That's really the invasion part. So any questions on that? Cool. What was that? It's interesting and intricate. So definitely. So anyway, here's just a more specific view of what we talked about if you want to get really into it. So this is the insulin receptor, which is a receptor tyrosine kinase. Just showing you the MAP kinase again. They're really showing the pathway and showing where RAF is. That when it gets mutated, it causes the activation of everything else. So main point: MAP kinase activation of new genes promote uncontrolled cell growth equals cancer. So that's really the main point I'm trying to drive home. And I kind of mentioned everything else already. So on so now knowing that, and also let's keep this in the perspective of drug discovery, right? In drug discovery, we are looking for first a disease to cure, right? We're looking for a disease target. Knowing all of the signal transduction that we just talked about gives you the knowledge to define a drug target, whether that be whatever protein it is. And of course, there's assays to validate that target. But once you have that target, we need to look at that target to see, can we drug it? Is it druggable? If so, where? That's what this is. So what they did after we figured out that BRAF is a big deal, we looked at the structure of BRAF. A lot of crystallization was done. A lot of genome work was done to figure out the sequence and how it could be manipulated. So here is what BRAF does is it gets activated by KRAS, and then it activates MEK, which is a kinase that's below it. So it basically takes one signal and brings it somewhere else. That's really all it does. So what can we figure out, and what if we were developing an inhibitor, what can we do? And this is something that all of you guys can answer. So if we're developing an inhibitor, right, you're looking at this. The protein of interest that is mutated has two domains that are important. One of the domains binds to the previous thing, and then it has another domain which takes the signal from the previous thing to activate the post thing, the post enzyme. So I'll ask you guys, what are two ways we can inhibit this protein? Anybody want to answer? You do one, you do the other. Yeah, so you can inhibit one. Either you can inhibit one as the target, or you can inhibit one by inhibiting the BRAF as the target. Both are okay. And then what were you going to say? Inhibit two. Right. So you can inhibit two, or you can inhibit the protein or you can inhibit two itself, or you can inhibit the binding site of BRAF that binds to two. So you can basically eliminate one of those interactions. So that's what's another positive thing about, or a positive thing about picking a drug target that is more downstream. Meaning it's not at the top. Usually picking but they have a lot of side effects because they are more upstream and they have a lot of interplay with other pathways, which means they have more off-target effects and more. If you inhibit it, it has more off-target effects. But if you're downstream, it will only affect everything that's below it, which is the gene transcription, which is the important part that we're trying to inhibit. So that's why downstream is a good target, and also because they're more druggable than upstream proteins. And the reason why is just like we saw. Just from looking at a really crappy diagram that I drew, we can figure out 4 drugs, 4 ways to inhibit this interaction by either 1, 2, 3, 4. We can make inhibitors. So that gives the drug discoverers a lot of room to structurally look at it and be like, all right, we in the 3-dimensional structure, we have to find some pocket, either in here, or in here, or maybe allosterically. Maybe it binds here and shifts this over, or shifts the structure over to look like this, and then that causes this not to bind. So an allosteric inhibitor is perfectly okay. So that's the next thing, is you want to look at the domains and see what's druggable. So here we have the domain binding domain. I should be close to the left. We have the RAS binding domain right here. We have the C1 domain. Don't really worry about that. And we have the kinase domain. So these are the 2 important domains for the RAS binding domain is right here, the before signal, and then bringing it to the after, which is the kinase domain. So yeah, that's basically it. And there's mutations, there's different isoforms of RAF, but we have BRAF is the main one that was drugged because that's the one that has the mutation. Yeah, any questions on that? Okay, cool. All right, so you can look at it as a different kind of a mutation mapping of the BRAF, which is what they did after they figured out, all right, these are the domains. Let's look exactly where these mutations are and exactly how we can stop them and what type of inhibitor we should make. So the reason why we have to do this is because we want to target cancer proteins. We want to target mutated proteins. So somebody can answer this. What is the only difference between this BRAF versus a healthy BRAF? And you don't even have to look at it. You can just, it's kind of a big arrow pointing to it. So what's different? There's a mutation. There's, yeah, valine, whatever, 600E, right? Whatever it is. The only difference is that there's a mutation in one of them and no mutation in the other one, in the healthy one. So how can this be used to our advantage? Well, we can make a drug that binds to the E, where it wouldn't bind to the V. That's good. Glutamic acid it has a charge usually, and it's easier to conjugate something to that than it is to valine, however you want to say it. That amino acid doesn't come off much. I keep switching my pronunciation of it. Valine. Yeah. But yeah, so you can base your drug discovery efforts on which mutation you're targeting. So there's that. But also, you want to be specific, but maybe not too specific. By that, I mean BRAF is a good case where 90% of the mutations that are important are V600E. But you can also see in this genome map, there's V600D, V600K, V600R, there's G466A, E, and V, and G646, E, and Y. And then also there's a couple other ones. So there are a few mutations that can affect the function of BRAF. Also, there's hundreds more that maybe don't. But there's these key ones that can cause cancer. But in this case, V600E causes a strict abundance of the cancer, meaning it's easier to pick which mutant you want to go after. But in some proteins, that might not be the case. For KRAS, there is G12C, G12D, and there's also Q61E, or Q61 is a bunch for Q61, but Q61 could be its target also. So, but G12C is the main one, but the other ones also cause cancer. Yes. I'm sorry, repeat it. Yes. Good. Okay, so yeah, the question was, can we use RNA interference to cure cancer, right? So of course you can. So because these mutations happen, where are they happening, right? We're only talking about drug discovery on the protein level, on the post-translational level, right? RNA interference is a great way of drug discovery on the transcription level. So we can develop a complementary RNA sequence, a short hairpin RNA that has the sequence mimicking the V600E protein and have it bind to in the nucleus to V600E sequence in the DNA during transcription, and then take it out and kill it. So it's tough to do that because you have metabolism, you have a lot of things affecting. So RNA interference is good on a even on like a mouse model it could work. Definitely in the cells it can work in the in vitro level, but getting it into humans is very difficult. So there's a lot of complications there. The delivery system, yeah. I don't really know too much about that, but it is difficult. So yeah, good point. Yeah, so I mean, all this, keep in mind, there's a lot of other ways to cure things, right? I mean, we're in the where we have CAR T therapy, we have a lot of different gene therapy. You guys are all gene therapy people, right? But the traditional and most effective current approach and the most traditionally successful approach is drug discovery by targeting the proteins. But new fields are emerging. CRISPR, for example, that's a very important, very powerful technology. But yeah, so we're doing this. Okay, so yeah, that's about it. Any questions for this on this? Cool. So we can also once we see it on the mutation map, the next step is like, all right, we're focusing in on, all right, which domain? We have certain domains that are important to function. Then we have, all right, on those domains, where are the mutants? We want to target the mutants so we don't target the healthy cells, which is hard to do. There might still be, and there usually is, let's just say this, there always is, an off-target. There hasn't been a cancer drug developed that doesn't have off-target to the healthy cells somewhat. By this, I mean, maybe you have the IC50 for the V600E for the BRAF drug to be 1 nanomolar, but the IC50 for the on the wild type form or the healthy form is maybe 400 nanomolar or 1 micromolar. That's a big enough difference that we can still use it. There might be nausea, vomiting, but no life-threatening conditions where all your cells stop growing. So anyway, that's why dosage is important too for these cancer drugs, because there's a limit to where it starts to really affect healthy cells. All right, so then we have after this is we want to look structurally at the X-ray structure if you can crystallize it. Some proteins are very difficult to crystallize, where you want to see the exact interface of binding that you want to attack. This is what gives you the idea of which compounds to screen. If you want to take an antibody approach, what do you want to do? What's the approach you want to have? So we can look at, we're not going to go crazy detail, but there's these. But V600. They might be next to each other. This is just one diagram from a paper. But here, let's just say it's V600. And you have this drug by Bayer 439006, that binds in that position. So you can crystallize the protein with a drug to kind of see the binding motif of that drug. That is very, even if it's not it's never going to be your final drug, but it lays down a scaffold that can be used to make more potent interactions nearby. So this drug might only be a 1 micromolar binder, right? Very weak binder. But we were able to crystallize it, or they were able to crystallize it, and then able to think of and make projections of other synthesized compounds that can bind better, because now they know where it binds. That's a very important tool in drug discovery, because right now, like in the project that we're working on, we have no idea where anything binds, because these proteins we're working with are very difficult to crystallize. We also have no idea where even the endogenous peptide, there's an endogenous peptide that binds to this protein. We don't even know where that binds. There's just so little known about this protein. So once we get this information, it'll be very helpful. I don't, but we outsource that to different CROs that do. But it's very difficult to be successful with that. I wish if I had to do it over again, I would be an X-ray crystallographer or mass spec. If you do X-ray crystallography or mass spec, you are getting a job immediately. You get any job you want. But because there's so little, there's so few of them, and also mass spec is just there's a lot of people that do mass spec, but it's just so sought after by everybody that you'll always have a job. Especially protein mass spec, that's a really big deal, very big deal. In my graduate class, whatever that's the mass spec class, I could take small peptide sequences and figure out the mass spec, take a mass spec and be able to match it to the right one. That's about it. But if you're really good at protein mass spec, you can take the entire mass spec of a protein and figure out its conformation from mass spec. And you do a 3D mass spec too. Different like Raman charts and all. There's a lot of stuff that goes into it. It's crazy. Yeah. Your question? Right. So crystallography is extremely difficult, and there's a lot of conditions you need to play with. It's usually at extremely high concentrations. The buffers are not physiological. It's usually you're dealing with milligrams of protein, which is a lot. Usually we deal with micrograms. It's like milligrams of protein. And usually crystals fall apart if they're not in the right buffer or if they're not in the right. There's different speeds of crystallization, and there's a lot of I don't know. I never did crystallography. There's a lot of parameters that can easily just destroy your crystal structure. Also, it's very expensive to do because of all the material. So, yeah, it's difficult. But if you get in a lab or somewhere where you can learn how to do it, definitely take that opportunity. Okay. Oh. So anyway, so here was the paper on the first BRAF inhibitor approved for late-stage melanoma called I can't even say this one. I just call it PLX 403. Vemurafenib. Yes, that one. Vemurafenib. Yeah. Okay, so the PLX, we'll call it the PLX. So the PLX compound, right here. So it binds to BRAF V600E, and it inhibits the mutated form pretty well, and really has not many off-target binding places. So it's pretty good. Yeah. So let's see and there's a structure of it too. So structural wise, there was always a common core. So the first PLX compound, PLX 4720, is one of their starting compounds. So the modification involved. So this is probably the one they crystallized it with, the PLX 4720. They have a different name here, but there's 2 rings. No, probably not. It's probably different. But anyway, they probably crystallized it with that one too, with the one in the blue. And they figured out the binding motif. Once you figure out the binding motif, you can identify a core, right? And the core here is the bicyclic ring here, the kind of benzimidazole kind of ring over here, and the ketone to the phenyl. That's kind of the core, and also the sulfur. That didn't really change either. But there's different offshoots. In this case, they added another ring to it, and it's basically the same other than that. But yeah, a lot of times you can add different offshoots. Yes. Yes. So right. Good observation, Daniel. So the question was the ending of the drug name, IB, right? What does that mean? I think maybe we went over it the first day of class, but you could have forgotten it. So anybody want to answer that question? What does IB stand for? IB. Yeah, antibodies is AB. But what is IB? Yes, exactly. Good job. So IB is inhibitor. That makes sense, right? IB, inhibitor, right? So if it's something like Well, you can't tell from the name what it's inhibiting, but for a lot of times you can, because they might just be. There's some drugs I've seen that are like, I can't think of an exact one, but it might be something stupid like BRAF-IB, or if the protein has a cooler sounding name, like an actual word, they might just put IB at the end. So I've seen that before, where you can tell from looking at the name it's an inhibitor of that protein. But yeah, so it's a major breakthrough. There's different compounds that can affect the MAP kinase. Since then, there's been different drugs. So we have BRAF inhibitors, MEK inhibitors, so that have all been FDA approved. And even after the BRAF inhibitors, other companies started to come out and put in a lot of money to finding more of these drugs. So here's a good question. So why, from a business perspective, right? Business. So why would we, if we saw a competitor company come out with an inhibitor that is really good against melanoma, it's not perfect, by far perfect. Like, by really good, I mean it can cure maybe 20% of people. That's what really good is in the cancer world, at least back then. So if we see that drug got FDA approved, why would we want to pour a lot more money into figuring out, into discovering either a RAF inhibitor, another one, or a MEK inhibitor? Why do we want to do that? Good. So yeah, optimization is one, but also what else?
Is it because it's more downstream?
Yeah, so MEK inhibitor, definitely because it's more downstream. Also think about patients, right? Like, what'd you say? Availability. I thought you said something else that was more on point. But what were you saying? Oh. Yeah. What were you gonna say? Good. So further downstream, less off target, less side effects. Because really, for this case, RAF and MEK only act here. But you're right, though. I mean, in general, you're right. Price is a big one. Yes, price. But we also learned about this. Different patients mean different. V starts with a V. What do you say? Variability, exactly. Yeah, that's the word I was looking for. Variability, right? We know a lot of drugs, or a lot of people have different variability in their receptors, in their gene expression, and everything like that. Also, in variability of their side effects, right? This BRAF inhibitor, the PLX compound, it may have cured 20% of people with BRAF mutation. No, it cured 20% of the cancers, but 20% of them had BRAF mutation. It might have cured every BRAF mutant, or most of them, most BRAF patients, but it may have side effects in 10% of them that are really bad. Even though it cured them, it may have had side effects. So it's a competitor. So let's say the GSK compound that came after it, the one like 11, whatever, that one may have had less side effects for some of those patients, which they would prefer it more. So it's just like, why would you make a different car if someone else made a car? Why? Because maybe my car is faster. Maybe my car has better entertainment system, right? Same thing with drugs. Not that they have entertainment systems, but I'm just saying they have different marketable qualities, such as, oh, this one gives you less nausea and vomiting. Oh, damn, this one gives you more, but it's better efficacy. So these marketing techniques are marketed to hospitals, to doctors, to it's really competitive. That's what makes drug discovery a great field, is that that other companies are competing to make better drugs. It's great because yes, they're making a ton of money doing it, but they're also saving people at the same time. So it's great. Yeah. Yes, that's a great point. So he said that, what was your name? Kyle. Yeah, so Kyle said that you may spend less time because somebody already did some of the work, right? Exactly true. If the PLX team, they came up with the first X-ray structure for BRAF in cancer drug discovery. They They docked their drugs there. They figured out a binding pocket. So all that work is done. So now it's just modifying, a lot of times it's modifying the compound a little bit to where you can get the same, maybe a little bit more advantageous qualities, maybe less side effects, or maybe it's slightly more potent. You save work, and as long as you don't violate their patent, you're good. Sometimes they may do the exact same thing. They just pour in $1 billion to take some of the market share away. And so people buy their drug at the same time, you know? So really it's a lot of marketing and competition. So yeah, that's good. So somebody I know actually had developed a ERK inhibitor, right? Their ERK inhibitor, ERK is down here. There is a GSK compound, so that inhibits ERK pretty well, and it's FDA approved. But this person and their group came up with a ERK inhibitor that is extremely similar. It doesn't violate the patent, but it's extremely similar. Now they're trying to publish this work because it's academia. They were trying to publish it, not make it a drug, because it wouldn't work. It would probably work, but it's too similar. You know, it would violate the patent. But they don't even. The journals are turning down their publication, saying that it's not novel enough, even though it's a different structure, and they have their own data on it. Right? They're denying the publication because they're like, look, you just changed a carbon and another carbon and oxygen. And yeah, it's a different compound, but it's still technically under the same class of compounds that GSK worked on. So you want to still be novel enough in drug discovery to where you're not violating the patent, and also you're creating maybe novel action. So let's say, and this is where a case would be different. Let's say you change one carbon, but they forgot to change that carbon and they never saw what would happen. They change the carbon to an oxygen, increases the potency by tenfold. But it's their job, these big pharma's job, to really dissect the structure and look at every single modification and to see what it does. They're synthesizing thousands of compounds in this class to really rule out everything, so that competitors don't pick up on maybe one thing that, oh, if you change that carbon, it cures every single cancer. So that's why some of these companies pour money into that. Maybe they can find a functional group that wasn't modified and that can cure a lot more, or definitely more potent. So that's another thing. But yeah, it's a crazy landscape out there, definitely. Okay, moving on. So another aspect of drug discovery for cancer, and more specifically for kinases. Now, kinases, remember, they phosphorylate, but kinases are all very similar because they use ATP and they break it down to get that phosphate, right? They have an ATP domain, an ATPase domain to break down ATP. In the selectivity part of screening these compounds and these drugs, you need to test the drug for binding to other kinases, because a lot of kinases have domains that are similar. So here's an example. Every kinase has this domain. This is the kinase domain. And the kinase domain consists of a phosphate bonding region of the sugar adenosine region. But if we're making a competitive kinase inhibitor, which the BRAF inhibitor is, we want to make sure that we are specific to BRAF only and not something else, and not another kinase. Maybe if we target a different kinase like BRK, and it's doubly as potent in BRK, well, good news, you got a BRK inhibitor. But bad news, you're going to have really off-target effects when it comes to treating BRAF implicated melanoma. So here you see all three BRAF isoforms have an IC50 of around 30 to 100 nanomolar, and BRAF V600E being the most potent. But, and in regular BRAF, so here's what I was saying before, you want to target the mutant BRAF, not the healthy one. And look at the difference between the IC50 of the BRAF mutant versus the healthy one. It's threefold off. This is done in a lot of replicates, so it's pretty reproducible numbers. So V600E, 31, BRAF regular, 100. So if your dosage is, if you want to consider your dosage in nanomolar, you never give it nanomolar. It's always micrograms per mil of blood or whatever it is. But if you want to consider it, or just milligrams per kilogram, let's say you have your dosage around 50 nanomolar, right? That will be that'll be inhibiting all of the V600E. It won't really inhibit too many of the regular BRAF, and it will inhibit CRAF, but CRAF is so less prevalent. BRAF is the main isoform. So CRAF is almost irrelevant. But it does bind to CRAF as well. And then a lot of you are thinking, okay, what about SRMS and AC1, right? It has pretty high potency. It might. But turns out these kinases aren't too influential in any phenotypic response. So they might be kinases that are just either vestigial, they don't do much, or whatever they do, there's other proteins that also do it. So it's not really that much of a big deal. And the only way to figure that out is by in vivo testing. There are so many proteins in the human genome, and even what we call this the kinome. The kinome is the map of all the kinases in the human body, and there's 580 whatever. I think 583. I don't exactly know the number. I'm not going to lie. If I just said 583, it'd make me seem more sophisticated, but I'm like, it's 500 something. Pretty sure 580 something. But there's a lot of them, right? So all these kinases, we can't go and test for all of them. They only tested 200, which is a lot, but they didn't test all of them. If they saw adverse, and these 200 were picked as they are the most commonly implicated in side effects. They weren't randomly picked. But the one, and also, maybe they picked like SRMS and AC1, they just ended up not doing anything. You know, even though they're being bound to and maybe inhibited, they're not doing it. That inhibition is not turning over to a phenotypic response. So, yeah. So then we also look at these in the cell. Of course we do. Before we go into in vivo models, once we make the drug and optimize it a little bit, doing some SAR, right, and trying to come up with the optimal structure, we go to the cell work. And this is a Western blot. So the Western blot can tell us, and we'll go over that as one of our assays that we'll cover for the technique part. But basically what it is, it's a technique that can detect any type of protein in a cell at a specific time. So let's say, well, in reference to a control. And it's all done by antibodies. So basically, you take 2 cell culture plates. You treat 1 cell culture plate with a drug. You treat 1 cell culture plate with DMSO or no drug, right? Just a dummy, nothing. You let those, you let them sit there for 4 hours. What happens in that 4 hours is a cancer cell, the level of gene expression of a mutated cancer or an activated pathway, meaning proliferation, will increase, will be high. Of a successfully inhibited, meaning drugged cell line, the phosphorylation or the activation of that pathway will be inhibited, right? Decreased a lot. So then you can harvest the cells, you can spin them down, you can break them down, and you can get all the proteins in the solution. Then you can treat that solution with, or you can put it through, you can separate the solution by SDS, by electrophoresis, gel electrophoresis, using SDS, but yeah, gel electrophoresis, and you can separate the protein bands. Then you can treat these bands with antibodies. These antibodies bind specifically. Remember I said how antibodies are really specific? That's why they're great for drugs, right? You can bind also for tools. You can bind it specifically to either phospho-MEK, ERK, or ERK, meaning activated or not activated or total, right? In this case, we don't have activated and not activated. We have activated, total MEK. The reason why we have total is because we want to compare the amount of phosphorylated activated MEK versus the total amount in that cell. So after that, after we treat them with the antibodies bind. then the antibodies actually have an enzyme attached to the end of the enzyme. Or, no. An enzyme attached to the end of the antibody. This is called a horseradish peroxidase. So it sounds, yeah, but what it does is it cleaves a substrate. And this substrate, once it's cleaved, there's several substrates. Femo something. That's what I use, at least. It's been like a year now. But you cleave the substrate, and once it's cleaved, it fluoresces. So you can put the Western blot, it kind of looks like a gel electrophoresis, right? So after you treat it with the antibodies, you treat it with as a primary and a secondary antibody. Once you treat it with the antibodies, put the substrate cleaves, fluorescence. You put it under a detector. The detector now gives you bright bands. These bright bands mean that, one, the antibody bound to that protein, so it's there. Two, the secondary antibody bound, whatever. There's antibody bound, there's that enzyme at the end that cleave the substrate, which means that not only the antibody bound, but we have the fluorescent detector that is now lit up, showing that the protein is there. So this is what we call a Western blot. And I can go over it in more detail next time we lecture. And I'll go over, this is a very powerful technique for detecting any protein, as long as there's an antibody for it in solution, or in a cell, not in solution, in a cell. Yeah, Western blot's the most common. There's a Southern blot for DNA, a Northern blot, right? Yeah, for RNA. So yeah, Western blot easily the most common. What? I mean, I've never done Northern and Southern blot. Are they hard? I think they're all. Mm-hmm. Right. Western blot, it takes a long time. It's not easy. I'll say it's not easy. Not the easiest assay I've ever done. It takes a while. But I mean, it's easy to learn, but it just takes a while. And if you mess up one step, you're screwed. And it takes, I mean, in terms of the drug treatment and then the growth and everything, it takes more than a day to do. So let's put it that way. There was 1 week I had to do like 4 or 5 Western blots. It sucked. I had to do them I had to overlap them in one. I did like 3 in one day. And I had to like because there's a lot of incubations, like a 4-hour incubation step, a 1-hour incubation step. So I had to like overlap them. It was hectic. That's one reason to not do your PhD, because you'll have days like that. And nights like that too. Yeah. Mainly out of frustration. Like I wasn't getting the data I was wanting. So I just like, I think it was a compound screen. And I was like, I knew that if I didn't find some kind of modulator, that my thesis is done, and like I would have to do a new topic and like spend another year. And I'm like, I'm not doing that. So I stayed up all night screening all those compounds, and then just getting more compounds to screen, and then I finally found a few. So and it worked. Otherwise, I wouldn't be standing here. Crazy how it works. But anyway, okay, back to Western blot. So what we see here from the data of the BRAF inhibitor, and this is a PLX compound, but they call it RG7204. That's just the inhibitor, right? BRAF inhibitor. And over a concentration gradient, they saw the inhibition of MEK and ERK. So to remind you, where do MEK and ERK occur? So we're inhibiting RAF. So to inhibit RAF, it means that RAF is still going to be there. Even it's going to be inhibited, but it's going to be there. So doing a Western blot with RAF wouldn't make any sense. There wouldn't be much of a difference between RAF with the drug and RAF without the drug. There'll be the same amount of RAF. But because RAF will be inhibited successfully, there won't be an activated ERK. The ERK will be unphosphorylated. So that's what we're looking for. And we saw that what happens is the treating out at the top, look at the top one first. I'll stay here. I'll do the math. So the top, we have the compound and then the concentration. This is in micromolar. We have 50 nanomolar, 240 nanomolar, 1.2 micromolar, 6 micromolar, 30 micromolar. Now, over concentration, you see, so total MEC is constant. This is the control. This is saying that, all right, at each one of these cell lysate samples, the same amount of MEC was there. Sometimes I've done a Western blot where the amount of the control protein varied. And sometimes you like to have another control protein, like a beta tubulin or alpha tubulin. There's a protein that's in solution that it's cheap to get an antibody for that shows up in every lysate, just so you have a standard. So having one standard, they probably had another standard, but it was cut out here. But you see that MEC is very consistent, so you don't really need another standard. So sometimes you have where the MEC level decreases, and also the phospho MEC also decreases. But since the control is decreasing, it doesn't really show that the drug is doing anything, and it might be a false positive. That's why we have to have the control. The controls are very important for every experiment. So anyway, the control is good. The activated MEC shows that it's decreasing expression or decreasing activation with increasing drug. That's great. That's good. And at 1.2 micromolar of the drug, there's basically no activated MEC. Same thing with ERK. There's no MEC activated ERK. And it's in LOX cells. There's a melanoma cell line. Yes. Good question. Good question. So yeah, common question. So why is ERK has 2 bands and MEC has 1? You want to guess? Go. Not quite. So think about how these proteins are separated. They're separated based on mass. And for ERK, there are 2 different isoforms of ERK. ERK 1 and there's MEC 1 and 2 also. But there's 2 bands because there's 2 ERKs that are very similar in mass that the antibody will bind to both, but one of them is missing a section. So yeah, that's usually the case for a double band. Because remember, so this is gel electrophoresis. This is the way the protein bands are separated. So the heavier ones are going to be at the top. The lighter ones are going to be pushed more by the electric field, and they'll be towards the bottom. So yeah, that's how. So these are, and also ERK is lighter. ERK is lighter. Well, that also has to do with amount. So there's technically 2 for MEC, but because there's MEC 1 and 2, but they're overlapping because their masses are the same. Some cases the masses aren't the same. Yeah, that's a very good question. And don't look into too much that the top band is decreasing and the faster than the bottom band. That's probably a thought that you were having also, but that's irrelevant. I mean, that's just, you might do this again and you might get the opposite. You might get them different. It's just a variability. Like Western blot, something about them. Yeah, they're kind of easy to do, but it takes a while. They are very difficult to get perfect. So they might have to reproduce this exact same experiment. Some poor employee, well, I mean, I'm sure you're getting paid a lot, so whatever. But I would do it if we were on this stage of a drug discovery project. I'd stay up all night and do it. But that's a lot of money. But yeah, so they might do it again and again and again. This is probably the best one they got. This is the one they put here, so it's the best one they got. The other ones might have been a lot messier. Some cases the bands look like this. It's like there's like haziness to them. They're not solid, you know? That happens sometimes. Sometimes it's like kind of blotted, like that. I see that a lot. And then you have to just keep redoing it until you get it perfect. So that's the thing about assays. You know, you can do the entire thing right. If the data doesn't look really perfect, you might have to do it again for really big applications. The exploratory stuff, not really, but when you're at the final product, you have to do it until it's perfect. But yeah, so and even in ERK, you can see that total ERK is the same, and then phospho ERK decreases with increasing concentration of the drug. Okay, any questions on that? No. Well, a couple more points here. So we're using these LOX cells, which are melanoma cell lines, which we need to be showing a constitutively active cell line. Now, your cell line choice in Western blot is really, but any Western blot of any drug discovery project, super important, because if you're doing HeLa cells, who's ever worked with HeLa cells before or heard of them before? Yeah, Henrietta Lacks. Yeah, exactly. So they are one of the most common cell lines used to date between that and E. coli. Well, HeLa cells are the most common mammalian cell line used. I'll say that. And the HeLa cells, this is a cancer patient in like the 50s, Henrietta Lacks. She had, well, this cell line, what cancer was it? I don't even know, but this cancer cell line, you know? Yes, it's the immortal cell line, right? So p53 suppression, but also with that, there's a ton of mutations. So you can get the mutation map for HeLa cells. There's a lot. So this would work on that cell line if there's well, I know this would work on the cell line because I know that my SOS inhibitors worked on this on HeLa cells. So then anything in the MAP kinase would work. But it's really important, the point is, it's really important to test on different cell lines. Because what if on a healthy cell line, you see this type of inhibition? That's not good. What if on a healthy cell line, you see activation for some reason, activation of downstream effectors? That's not good. It might cause cancer. You might have some weird reaction that you're not expecting. That's why, like, a lot of the science has to do with disproving other things from happening. Not really disproving your drug works, but disproving it works on other cells, other patients, other demographics of people, etc. You know, because yeah, that's really something. Yeah, that's how it works. Okay, cool. Another thing I want to mention. I know I'm on the slide for a long time, but another thing, last thing. We mentioned that the drug has an IC50 of 31 nanomolar. Yet, in here, if we had to guess the point of 50% inhibition going from 50 nanomolar to 30 micromolar, the 50% inhibition point, if this is 100% and this is 0-ish, 50 is probably around here, between 240 nanomolar and I'd say around 240 nanomolar. That's around 50%. So why do you think that the affinity has been greatly reduced from 30 nanomolar to 240 nanomolar, going from here? I'll give you a hint. This is a FRET assay or some kind of other enzyme assay. It's an in solution assay compared to this Western blot. So why do you think there's a difference there? Whereas the drug is weaker in the Western blot. Tough question, but think about, so I'll give you a big hint, is that this is in the cell. So why would a drug be weaker when treated on a cell versus in a solution in an enzyme setting? Right, good. So that's basically it. So it's a lot of the pharmacokinetic stuff that we learned, right? But on a smaller level. The cell is a living body, right? To get to the cell, to get to the target of the protein of interest, the action site, the drug needs to, one, transverse the cell membrane. What percentage of them will transverse the cell membrane? Who knows? Number two, needs to find the protein of interest. Needs to find BRAF. There's thousands of proteins in the cell. How is this drug going to find BRAF? It might have off targets. That what they do is, once this drug binds to the off targets, it reduces the effective concentration. So you might have 240 nanomolar getting in, but 32 nanomolar is actually binding. That's what the IC50 is. So that makes sense. But in a cell, usually the affinity is greatly reduced. Well, greatly meaning 5 to 10 times reduced, comparing a in solution assay versus the cell assay. So that's a really important thing to know, because even with the best drugs ever, you see these Western blots are usually in the high nanomolar or low micromolar. So that's, and then meanwhile you look at their enzyme inhibition data, and they're in the low nanomolar or even high picomolar. So that's why it's transitioning from the cell or from the enzyme setting, the in solution setting, to the cell setting. That's why using low nanomolar as a standard is really good for in solution assays, because we know that at worst they'll translate into the cell at low micromolar, which is still acceptable for a drug. So yeah, and to compare my thesis, it was a lot of skepticism going into my drugs for MAP kinase, because we had IC50s around 20 to 30 or 10 to, my best one was 19 micromolar. 19 micromolar. Here we're at 31 nanomolar. Of course, it's FDA approved, whatever. But still, the 19 micromolar. So my advisor is like, look, if you get this thing to do anything to the cell, that will be miraculous. Because we're expecting, if it's 19 micromolar, we're expecting it would have to be millimolar to do something. And you can't treat a cell with millimolar of the protein or of the drug. It will usually lead to cytotoxicity. And you can't even get that high. You can't even get that high. But my Western blots were pretty damn good. And I got the same affinity, which is really weird. Very similar double-digit micromolar affinity in Western blot and in the solution. The same affinity. Usually I said, what I should say, it's greatly reduced. I got the same. The reason, and it took us a lot of time to figure this out, was because, all right, it's probably because my compounds were very good at penetrating the cell. And even though their affinity wasn't great, they didn't bind at nanomolar affinity, they were very good at selecting the target. They're very good at getting to SOS was my target. They're very good at getting to SOS and binding to SOS and not anything else. So, or I could have been completely wrong. Maybe they bound to other stuff that also caused the same response in MEK and ERK. Maybe. But I don't know. There's only so much you could do in 4 years. Yeah. Yeah. I mean, for this field, I mean, PhDs usually last 6. 6 is about average. 5 to 6. But also, I was there as an undergrad, so I was in the same lab as an undergrad, so that helped it a little bit. Yeah, basically. But I had a different new project, but same advisor. So we worked well together. Okay, so we're almost done actually. Kind of. Yeah. Yeah, thanks. Thanks for counting. So anyway, we also looked at when these drugs come out, we also want to see their range of efficacy in different types of cancer. Now, when you make a cancer drug, I get asked this question a lot. I mean, I haven't made a cancer drug. I don't want to talk like I've made a cancer drug, but when I tell people that aren't in the scientific field, like friends, family, stuff like that, that I do cancer research, which I did because my thesis and everything, right? It's cancer research. So a common question that you will get if you're in cancer research is what type of cancer, right? It's now knowing what we know, it's very difficult to answer that, because these are the different types of cancers that the BRAF inhibitor was effective against to different degrees. This was FDA approved for melanoma, but very frequently there are cancer drugs that are FDA approved for not just melanoma. They maybe start out as melanoma, but then they say, oh, more post-market trials show that they have clinical efficacy against lung cancer, against breast cancer, and maybe pancreatic cancer. So they might be approved for other cases. So it's tough to answer. And since I was working on KRAS, I can't really answer which kind of cancer I was working on, because KRAS is involved in pancreatic, involved in lung, involved in skin, involved in breast, involved in a lot, colon, right? So prostate. So yeah, I don't really like that question of which cancer. But anyway, so let's look at the efficacy of BRAF mutants based on IC50. So on the left, we see on the left y-axis, we see IC50. The lower, the better, right? So again, mostly melanomas, different types of melanomas in different same type of melanoma, different cell lines. That is what is on the x-axis, is different cell lines. So here's the LOX cell line, the one we were looking at, LOX, right? Yeah, the one we were looking at before. And then here's other ones. So a different range of melanoma cell lines, we see a low IC50s or high affinity. Much else, not really. But this rare, this breast cancer in this MDAM or whatever that is, 1B435 cell line, we see a really good IC50 there. Why is that? I don't know. That's just the way it was, the way it worked. And then maybe that cell line has a huge implication, or BRAF plays a huge role in the uncontrolled proliferation of that cell line. And so then it could be approved, bless you. So then it was probably approved for those cell lines, for people who have very similar genome to those cell lines. But for the other cancers, not really. And in some cases, there's overlap. But here in this BRAF mutant, we don't really see that. Any questions there? Cool. So we also need to look at. So this was the different. This is like basically the accumulation of the Western blot, right? And we can deduce the IC50s from the Western blot. Then you want to go into in vivo models. So the in vivo models, we use mouse and we use tumor volume tumor growth assays or growth inhibition assays, where you treat you treat with the drug, and then you look at tumor volume over time. So you take mouse A and mouse B. Mouse A you both give them cancer, or you don't give them cancer. You breed them with V600E, so they are born with cancer. Then you wait till they get a certain age, or the same age, and then you treat them with a drug or a placebo. The placebo in black, over time, over day 6, 8, 14, 18, the tumor size, the tumor volume will increase, right? Because cancer is growing. In the drugged candidate, the drugged mouse, the tumor volume will stay the same or decrease. That's exactly what we saw. That's a huge separation there. That shows that in vivo, the drug is great. It works. It reduces the tumor size, or it leads to the. Well, yeah. I mean, you waited until you have a tumor first, very small one, and then it decreases the tumor size. And then you see that again. Yeah, it completely. Following these studies, Roche started clinical trials to see if the drug would work against melanoma cancer patients. Yeah. So yeah, and you see that here for this other compound as well. Are these both? Yeah, these are both the same compound but different dosages. And for a higher dosage, 20 mg/kg, milligrams per kilogram of body weight, you see a complete inhibition, a complete reduction of the tumor. That's great. That means that this drug is really something. But we see clinical trials in humans. That's the obvious last step, right? We talked about different types of clinical trials. But we can see a PET scan, and you can look at the other scans later, but PET scan, baseline, somebody with different tumors, everything darkened are different melanomas. So you have a lot of melanoma on this patient. Day 15, a lot less, 95% regression of the tumors. So that's incredible. This is a really groundbreaking drug. So yeah. And tissues show a decrease in ERK phosphorylation. Yeah. So but yeah, that's basically, is that it? Oh yeah. So any questions on that? On the clinical trials? And then we see tumor shrinkage. So we also want to look at this is more data from the clinical trials, not just looking at one patient, but looking at a lot of different people with different stages of melanoma. This is approved for late stage melanoma. As you saw, this person had a lot of melanoma, and in 15 days, it was almost reduced to nothing. Then these lighter spots might be just the organs, the outline of organs. That's probably the heart. But there was really not many tumors, or nothing really left. But yes. It's not on here? Yeah. I don't know. It's probably the scan. I don't know. Might be the scan. Like here, the brain's darker. So it might be everything was just darker. I don't know. It's a good question. Yeah, I saw it too. I was like, I don't know. But yeah. Yeah, it goes to the lymphatic system, like these dead cells, and they just it wouldn't be that much. Yeah. Might be the spleen. I don't know. I'm not an MD. Right, right. Yeah, I mean it was just a different scan. But they should be the same layer because you want to look for the because the melanoma, think about what layer it's on, very surface layer. Right. Mhm. It's forged. They fake the data. Yeah, I don't think they faked the data, but I see your point. I'll look into that more actually. I'm really curious. I have the source. The source is right here on the bottom. No, we can look at it. What are you confused about? Well, yeah, yeah, yeah. So the shadow is usually the and so here on the bone is the tumor. Yeah, and then the lung, the tumor's right there, right where the my right there, small. Yeah, right where the arrow is. That's the tumor. You see it decreases over time. Yeah, it becomes like very, very small. Right where my arrow is now, that's where the tumor is 8 weeks, right there. The little tiny dot. Yeah. And there's another one actually over here on the right, which it becomes almost nothing. Yeah. We'll look at that. Cool. Oh yeah. So the last thing we want to test over different people and different tumor sizes. So we have each one of these bars is a different melanoma patient with a different stage. The stage is depicted by the color. The time of the response is with different bars, but basically over the course of several months, we see so the diamond in each of the bars shows the total decrease of the tumor size, and the bar length is the amount of or is the month of the is the progressiveness of the disease, and then you see the time. Oh, the time of the time of partial response. So the star is when they start taking the drug, and the different melanomas over time. So I guess this is showing that they take them for a long duration. And then here on the well, we really pair that with the left, and we see the decrease. So you can compare the B graph to the A graph and see that these may be the same patients. And you see in that time frame, you are seeing a decreased size of the tumor from the baseline, from the original size. So you had two people where it actually increased. That's where the other drugs might come into play. So that's why you want to make other competitors to this drug to maybe turn those cases into positive cases. So, yeah. All right. So basically, conclusions is you have BRAF mutants lead to constitutive growth, lead to active MAP kinase signaling. You have the PLX4032 is the first BRAF inhibitor that's approved, and it treats a wide range of melanomas at later stages. But yeah, so the important thing and the tough thing about treating cancer is that cancer cells, once they get to a certain stage, they can switch pathways based on the mechanism we talked about. And that's why recurrence of cancer is likely, because there's an upregulation of these other pathways, PI3K and other things, that can lead to constitutive growth in other pathways, so in the person. So that's why if you have different, that's why you need to have different regimens of chemotherapies, and they always come up with this for every patient, that